Hello everyone. It's my pleasure to present McDevol in a microbiome GOSI. McDevol is a method for probabilistic metagenome binding using Bayesian statistics. Following an excellent talk from by semi-bin developer, um, so this introduction, metagenome binding is basically a solver for interesting puzzle where it takes a metagenomic context and without knowing how many genomes and how abundant each genomes are in the sample, it can reconstruct microbial genomes from the context set that we refer as a metagenome assembled genomes. We know that microbes are everywhere. They exist as a community that we refer as microbiome. Analysis of environmental samples through next generation sequencing followed by assembly and binding results in MAX. MAX represent genome information of microbiome, which helps us to understand diversity and metabolic potential of microbial community. Hence, great effort like Magnify DB catalogs max from different environment to enable diverse application of max in different fields like finding gene clusters in microbial genomes using space dust. For analysis of application of max, they are assessed based on two criteria. One is how complete and how pure they are, generally using based on single copy marker genes. Incomplete max result in lack of information, while contaminated max result in misinformation. So for accurate downstream analysis, one need to have a high completeness, especially high purity max. In computational pipeline, binding constantly evolves with the time, starting from 2004, where KMR information was first used to bind contigs, and there are many methods during that time developed to improve the binding based on KMR frequency. Later, people realized that contig abundance, which is the number of reads aligned to the contig, can improve the binding. The reason because the contigs comes from this same genome would have a correlated abundance profile across samples. So when we combine contig abundance with the KMR frequency, we get better binding results, and many famous methods are built upon that. Recently, people also integrate codon counts, read linkage, assembly graph, etc. to improve binding. Also, recently, neural network entered into binding field to improve binding, thereby uh, improving the quality of max. Most of the cases, KMR frequency and the contig abundances are compared in different way to compute distance between contigs so that we can cluster robustly. We have come up with a novel distance measure using Bayesian theorem. Let's say the key idea is that for a given contig, we observe certain abundances in each sample. Here I illustrated with the three samples, and the abundance profile follow a multinomial distribution. Here the corners represent samples and contour represent the probability of observing counts in each sample. The idea is that our distance compares this multinomial distribution between contigs to find similar contigs. For this, we have a generative model that makes use of multinomially distributed count profiles of contig x and contig x prime. Here, the count is a vector where it comprises of abundance in each sample plus KMR frequency of 256 KMR types. And this generative model helps us to calculate likelihood of contig x and x prime having a same latent variable. Same latent variable means they belong to the same genome. And this likelihood helps us to calculate posterior probability using Bayes theorem that contigs x and x prime having their count profiles drawn from the same underlying distribution. We get distance by taking negative log of posterior probability. Shown here is the performance of our Bayesian distance. The distances are projected onto the UMAP, and the darker blue color indicate contigs comes from the same genome. And you can see that Bayesian distance able to get one compact cluster for contigs from one genome, while Euclidean distance and Pearson correlation coefficient, as used in other methods, have a dispersed clusters for a given genome, suggesting Bayesian distance that we have developed is more robust for clustering metagenomic contigs. We apply Bayesian distance in a simple agglomerative clustering, where we first sort the context based on its total abundance in a descending order, so that we take context with has a very large high abundance profile. We compute Bayesian distance between the cluster centroid and all the members in the data set and find the closest member using d0 distance cutoff. We find clusters in a similar fashion in an iterative manner. In the in the subsequent iteration, if a cluster centroid finds a member that is already part of pre-cluster but has a shorter distance than in the previous cluster centroid, we update the cluster association of that member. In this way, we obtain clusters with a complexity of context times clusters, which gives us a speed and memory efficiency. However, these initial clusters are not well in recovering genome with a good coverage. For this, we apply density-based clustering. The key idea is that cluster centroid should have a higher density than its neighbor points, and also it should be far from a next higher density point by a large distance. And we compute Bayesian distances for this clustering to get density-based clusters. With this, McDevol workflow starts by calculating contic abundance from alignment BAM files and Kramer frequency from assembled sequence file. Combine these two count profiles, and we compute Bayesian theorem-based probabilistic distance on the fly during our agglomerative clustering. The initial clusters we get from the clustering would then be refined by density-based clustering. Beans comes out of these two-stage clustering are the output from McDevol. Shown here is a McDevol performance on CAMI2 datasets, marine and plant associated. And box plot represent purity per bin in percentage. Vertical yellow line indicates average purity. And we see that McDevol achieves highest performance in purity compared to MetaBat2, MaxPin2, and Concord. They are all famously used binners. And we also happy to see that McDevol achieves high purity as high as WAM could achieve, which applies a neural network. Now I have to compare with the semi-bin2 also. Um, Shown in the middle is basically average completeness per genome in y-axis and average purity per bin. And we see that McDowell achieves comparable or even better completeness compared to MetaBat2, MaxPin2 with a very high purity. Another more important aspect to look at is how many good quality bins we get from the binning. For this, shown here is AMBER score greater than 90, greater than 70, and greater than 50, which is a combined score of completeness and purity. We see that McDowell provides more reasonable quality bins than other binners that we have compared for this benchmarking. We also see that McDowell can recover circular elements like plasmids and viral genomes. Shown is a bar plot for various AMBER accuracy quality measures, and we see that McDowell outperforms Concord, and the performance is closer to WAM, which is the current best tool in recovering circular elements that was tested in CAMI2 assessment study. Shown here is the integrative binning results. Generally, MetaRap in combination with the MetaBat2, MaxPin2, and Concord is widely used in major application studies. And the performance in terms of completeness and purity in y-axis and the number of bins in x-axis shown. Red color indicates the canonical combination. And we see if we apply McDowell, the performances improves. We get more bins in terms of completeness and purity. With a high completeness and purity, with more bins have better completeness and purity, suggesting that McDowell improves integrative binning. And we know that MetaBat2 is best in terms of times and memory usage. And we see that when we look at peak memory usage, McDowell requires lower peak memory than MetaBat2, and time is also comparable. In summary, what I say that McDowell is very simple. It resource efficient binner that provides more high quality bins, thereby suitable for large scale metagenome data analysis. And McDowell can be considered as one of the good choice for integrative binning. The tool is freely available in GitHub. You can use the QR code. Shown here is a UMAP. This is from my test trial runs. It's not useful for my work directly, so I made use of them for this art purpose to thank ISMB and ECCB plus MICROBIOME GOSI organization for giving me this opportunity. With that, thank you for listening, and I'm happy to take any questions.
Question?
Thanks, great talk. And if I understand correctly, you have a hyperparameter alpha in the Dirichlet distribution. How do you tune this one for your distance?
Yeah, so we actually apply optimization procedure where we have an observed count and we find the alpha that maximizes our probability of observing the count.
Yeah, second question. You split it into two steps. One is calculate this alpha, second one will do the class tree, right? So in my mind, it will be more appropriate to consider that there's some kind of latent class P that is discrete P1, P2, P3, and then optimize this one into several latent classes.
Yeah, we actually fine. So we optimize alpha and then we actually calculate the P1 to Pn based on this sample.
Yeah, if
or
this
for
one combine
types.
into one step, just use a second something like EM algorithm to just use a soft partition, just like Gaussian graphic model, something.
Okay, that's an interesting idea.
Yeah.
I will discuss with you
Okay,
later.
thank you.
Yeah.